Okay, thank you for the introduction, and I'm happy to be here. And what I will do is to give you a sense of uh, some notions in immunology and some infectious disease, and let's just keep it as a two-way interaction. I think that would work better for all of us. And the topic that I have for today's discussion is capturing bugs using nets. Okay, we'll expand on this theme a little bit as we go along. And so basically, I understand that all of you are BS students, and so therefore we will try and walk through a little bit more in more detail so that we can. And please feel free because I don't teach BSc students, so I may be a little bit faster than what it would be, but I'll try to walk through. And there is no syllabus I need to cover. Okay, okay, so there is no rush. We'll cover whatever time permits. Okay, so essentially. All of so I come from the National Institute of Immunology, as was mentioned, and we do research in whole bunch of areas, not necessarily restricted to immunology. Obviously, immunology is a major focus area, but if you people might want to visit the website, and it's on Saturdays or Sundays, you are welcome to come there as well. It's a nice place to just visit. The rest of it, of course, is nice as well. Okay, so all of us are aware that we have, a lot of infectious diseases floating around, and we have a system in place that will help us protect against these various infections. So, and that is what is called the immune system. And as you would notice that pathogens can be of a variety of kinds. You have, bacteria. Right, I'm sure some of it you may have read as a part of your BS or even high school district. Pathogens, which can be bacteria, parasites, fungi, viruses. Virus का भी सब जान गए, right?
Yes.
All right. Yeah, COVID. How many of have had the pleasure of having COVID? Very team matter. Okay, all right. So essentially, and the immune system, the way it works is that it depends on how there are virus, for example, would be infecting a host cell. It replicates within the host cell because it requires the host machinery. Okay, for all the various processes that are involved in the replication. Whereas all the other ones are also dependent on the host in some ways, but not to the same extent. Now let's look at bacteria. Today we will be focusing on bacteria. Of course, as you are familiar, parasites are there. Fungi also can cause various diseases. So depending on whether a pathogen is an extracellular pathogen or an intracellular pathogen, the host system sees it differently. Okay, the way it is handled is different. For instance, and the definition of extracellular and intracellular is basically based on the fact that that particular pathogen is capable of replicating outside any host cell. I mean, viruses for sure cannot replicate outside the host cell, but you can have bacteria. For example, Streptococcus pneumoniae is an example. There are many other examples out there. Okay, those are capable of replicating outside the cell. Now, so what we will do is that we will use Streptococcus as an example to illustrate some of the things that we are familiar. And the notion with which this has been prepared is that some of the concepts that might be applicable across whole spectrum of categories of pathogen. And again, just to make it easier to understand, we are taking one particular example, Streptococcus as such. Okay. Now, again, I'm not sure how familiar you are with the overall immune system, but just to introduce some of the players that are there involved in this scheme of things. So it's a quite elaborate system with lots of different immune cell types that are there. Okay, and I will introduce you to some of the players. All these various immune cell types actually develop in the bone marrow. In the adults, all of them have their origin in the bone marrow, and like any developmental processes, there are lots of different stages and steps involved they are all very well coordinated. Otherwise, you would be a lot of trouble in general. Okay. Now let's think about this. So as I said, this all you start with a kind of stem cell in the bone marrow, and they would give rise to lymphoid cells. You have other types of cells that are there. I'll just to keep it simple, I will not go into all the various cell types. You have something called B cells, which are important for generating antibodies. Okay, now all of you have, there's been COVID has brought immunology to everybody's home. Okay, so antibodies, RT-PCR, immunity, all of these are household names now. So B cells are the ones which, on further differentiation, would produce antibodies. Okay, so just keep that in mind. In addition to it, you also have various cell types. Okay, there are T cells and natural killer cells and dendritic cells. These are all, all of them have their own biology, and these are all huge sections in immunology. Okay, now another cell type that I would like you to remember is something called phagocytes. Okay, remember when a host is invaded or infected with a pathogen, and remember we are focusing on bacteria at this point in time. So there are a lot of different mechanisms that would work, but one of them involves identifying the pathogen, and it is taken up by what are called phagocytes. Okay, they would take it up and then they would kill it within the cell. Again, many mechanisms involved, but they would be killed by the bacteria. So one of them is called macrophages. Okay. The other cell type that is involved in this process is called neutrophils. Okay, the neutrophils these are in human blood, for example, the most abundant cell type is neutrophils. Okay, 70% plus. Okay, so they contribute to these processes, and as you can see, there are many others that I have not touched upon. Just to let you know that it's an elaborate system which has multiple dimensions, and for any immune response, it is not that one cell does everything. There is cooperation between these and coordination between these different setups. Okay, so just to keep that in mind. Now, as I was pointing out, I thought it would be a good idea to use one example as a case study, so to speak, so that because each of them will have their own ways of looking at it, and they would have their. If you are talking about, let's say, TB, the biology changes. It's also a bacteria, but the entire process is different. The components of the immune system that contribute to them are also different. So just to keep it, focused, we will not get into many of the other aspects. Okay. So this is just to get you oriented a little bit. It's like the SARS-CoV-2 virus. This is also an airborne infection, and once it goes into the nasopharynx, it can establish. It stays there as a common cell, meaning in all probability, all of us carry this at this point in time. So once you have immunocompromised, maybe for example you had another viral infection, your immunity comes down, then this pathogen can take advantage and invade, meaning get into the bloodstream, get into the lungs and stuff like that. The normal place where it resides is the nasopharynx. Okay, it's up here. Okay, so under conditions, so it's basically asymptomatic. You would not be showing any symptoms whatsoever, but you will test positive if one were to sit down and do the test. Okay. Now again, I will not go into the details in the interest of time, but essentially under conditions which are not necessarily well understood, they can go to other organs, for example, lungs. That is where the name Staphylococcus pneumoniae comes in. And from blood, it can go to the bloodstream, causing bacteremia, or also called sepsis, and also it causes meningitis. Okay, so it causes variety of diseases. But just to let you know that, for our purposes of today's class, the pathogen has invaded the host, and now the immune system has the job of trying to get rid of it. Okay, so we will, as I said, we will be looking at pneumonia as one aspect subsequently in our studies. Okay, now this is just a cartoon representation. The way the bacteria looks under the microscope is that it is an encapsulated bacteria, which means that other than the body of the bacteria, you have a polysaccharide-based capsule, which can be very, very thick. It's like you will have the bacteria, which is this. If you imagine this as the bacteria, this entire table would be how it is going to show up, and this is all carbohydrates. So it's a huge jungle around the bacteria. Streptococcus pneumoniae is not the only one. You have other bacteria, encapsulated bacteria. A lot of these things that I'm going to be discussing with you today would also apply to, let's say, Haemophilus influenza or even Neisseria meningitis. These are also encapsulated bacteria. So the essence of this particular cartoon here is that you have this is the outer one is representing the capsule, and it is, as I said, it's basically polysaccharide based, and so because it's a huge jungle, few proteins can also kind of jet out and show up, on the surface. Okay, why is this important? But for example, in this particular set of class of organisms, the form of protective immunity, we'll elaborate on this in a bit, is antibody mediated. What that means is that in order for the pathogen, the host to clear the infection, you need the antibodies are going to be the main instrument used by the immune system to get rid of it. How it does, we'll look at it later. Okay, but fundamentally, what we are talking about is that there are several other proteins which are also contributing to this pathogenesis. So remember, just because bacteria, you can have a wound, you don't know, it could be any bacteria that can come. Not all cuts are cause infection and spread. Okay, some of them are just not capable of causing any disease at all. Okay, so let's then look at, as I said, we have polysaccharide, and there are lots of other factors. I will again not go into it. And in our lab has been trying to understand what are the various proteins which actually are responsible for the disease. Okay, because if you say somebody had pneumonia, okay, now how did the pathogen bring about this pneumonia? Hua kaise? Because obviously we know outcome. You go to the hospital, they will say X-ray karo, it's full of fluids, and obviously you qualify as a pneumonia patient. Okay, so we have been looking at this in our scheme of things. So one of our students said, okay, while all of us, at least most of the lab has been looking at, what are the proteins on the surface which are contributing, or virulence factors as it is called. So why don't we look at what is being released by the pathogen? Remember, pathogen can also cause other aspects. You must have heard about various toxins that are there. Anybody who can give me an example of a toxin released by a bacteria? Yeah. Okay, so some of these toxins are the ones which are responsible. Let's say for tetanus, as an example, the toxin. If you just inject the toxin into the host, you will get all the symptoms that the whole bacteria will create. Okay, so that they are that potent, and that it can contribute to the entire disease phenomenology completely. Okay, so this bacteria also releases what is called pneumolysin up here. Okay, so other than that, if there is anything that is new that might be there, is what was her point. Okay, so we said, okay, let's go back and try to understand that. But now, as I was pointing out, for this class of pathogens called encapsulated bacteria, the main mechanism used to fight the pathogen, to clear the infection, is what is called opsonophagocytic clearance. Okay, which essentially means that, for example, as I was mentioning, the antibodies are a key factor. So in the presence of the antibodies, the phagocytic cells can recognize the pathogen, and then the antibodies facilitate the uptake of the pathogen, and it is taken up. Okay, there are other opsonins as well. For example, complement system. Okay, so essentially for the responses against, for example, a bacterial protein on the surface or elsewhere, this is the B cell, which has antibodies on the cell surface, and it is recognizing the bacteria. Okay, some protein, for example, on the bacteria. It does not necessarily have to be protein. It can be anything that is foreign as far as the immune system is concerned. But for protein-based antigens, you need some help from another category of important cell types called T cells, T helper cells specifically. And there are some interactions that happen. We will not go into it. All of this we will treat it as help from another cell type. And this B cell will undergo differentiation and it will become an antibody forming cell. Okay, another name for antibody forming cells are plasma cells. Okay, so they are the ones which actually release this antibody, and this antibody, remember, it was recognizing this red antigen on the surface, that these antibodies are going to be in your bloodstream. They will bind the bacteria, and the one aspect is that obviously if you have, let's say, in the nasopharynx, it has to get into our body. Okay, as far as the immune system is concerned, nasopharynx is outside the immune system. Okay, so if you have to get in, obviously the bacteria has to first adhere to some cell type. Epithelial cell lining is all over the place. So if these antibodies prevent the bacteria from attaching to the host cell, then it would not be able to invade or get into the host. So therefore, it is kind of preventing this from happening. There are, as I said, there are also phagocytes. What were the names we said? Macrophages and B cells, neutrophils or macrophages. Okay, so so I'm just using phagocytes as a combined term for all of them. So this is they have some receptors which can recognize this antibody coated bacteria called Fc receptors. Okay, so just remember that there is some mechanism of it. Okay, that's the key thing here. And they are taken up and then not described here, but there are processes within the cell that would kill the bacteria. You could also have something called complement. Okay, this can also help the uptake better. In addition to it, it could also help in the uptake. So these are both complement and the antibodies which serve as what are called opsonate. Both put together, they would also result in the uptake. So essentially, the antibodies are contributing to the clearance. Okay. Now this is how pretty much most of the stuff happens. What I will do is I will walk you through the journey that Hina in the lab took, and as it panned out. Okay, not the way we would put it in, so that the way you face it in the lab on a daily basis. Okay. So this is just to let you know that the neutrophils have their mechanism. Some of them are described, so you don't have to get overwhelmed with what is there. But essentially, the microbe is picked up. There is phagocytosis. It goes into the various phagosomes. I'm sure you're aware of this. And then there are components within the cell which are kind of thrown at the bacteria, and those are killed. Okay. Now, as I said, Hina in the lab was trying to understand what are the proteins that are being released by the bacteria, and whether any of those contribute to this pathogenesis. Okay. Originally, as I said, immunolysin is already aware. Okay. So as a research student, you are trying to create and understand new knowledge. Okay. So if that is done, then we don't spend a lot more time. Some people elsewhere in the world have already looked at what is other things that you can help with. So that's the notion. So how do you do this experiment? Okay, you culture the bacteria. Okay, so this is I'm sure so many of you are in different labs, and let's say E. coli, for example, you have a broth, you shake it or grow it. So similar process happens for the bacteria, and so when we are saying what is being secreted, that means that set of proteins would be there in the culture medium. Okay, so what she landed up doing was she pulled out the cells pellet कर लिया. Everybody is okay with Hindi?
Yes.
किसी का नहीं है तो बता दो.
Anybody
Okay, not to
she's attending online.
Okay, all right. Okay, so I'll continue to do that. So essentially, what the idea here was that you pellet, and what you get in the supernatant is a collection of what is called secretome. All the ones that are thrown out, it's a collective term for everything that has been. So now, and again, I'm not showing here, but how do you find out? How do you know what are the proteins that are there? So
This bacteria is not infectious, right?
it is infectious.
So how are they handling
This is done in a biosafety level 3 lab.
Level
Yeah, level 3 lab. So you when you go in, you will appear to
Yes,
them.
you also understand. Because we are also culturing, they will think like it is so easy. Not that it is negative
No, so
pressure,
if when you see the student going, you will appear that he's a space में जाने वाला है.
Right.
One suit for the entire thing, goggled up, and in addition to whatever other things, so you have just eyes and everything is, and the lab has 3-4 levels of negative pressure, and there are, I mean, it's a very expensive lab to run. We spend more than 1 crore for electricity, forget about everything else on the lab. So it's a I mean, specialized lab in a sense. Okay, so you are right. Okay, so essentially, so the culture that is, so the supernatant was taken. She concentrated the supernatant because then you have all the proteins in a smaller volume, and do what is called two-dimensional gel electrophoresis. Okay, so there, some of you may have seen some agarose gel or SGS page and stuff like that happening, or I'm sure some of you may have even had the opportunity to do it. So this one happens is you do it in one dimension, like you separate it on the basis of size, and you separate it on the basis of PI. Okay, another property that can be used, and you have it spreads out on a sheet. Okay, otherwise the lanes that you see will be in one line. So this you do it that way, and then you spread it another way. Okay, using another property. So this, okay, so it turns out to be the more than 200 different spots that you can see on those.
So what is this mutation?
I'll just come to this. Okay, so essentially what was happening is that she was using all kinds of different assays to figure out, I mean, what could be a toxin we don't know. Okay, so you have to have some way of finding out whether this kind of toxin is there, this kind of thing, and she was doing. So it turns out that in her scheme of things, in the supernatant, she finds that there is a DNase activity. DNase is an enzyme which digests DNA. Okay, so if you, some of you may be using restriction enzyme, it is a kind of DNA. Okay, so the point was, I mean, so she was very surprised. Remember, normally you have DNA within the cell. Okay, DNA is within the cell, and there are a lot of different kinds of cell new DNA synthesis is happening. There is also breakdown and elsewhere. So you need DNase within the cell. So when she found that there is DNase activity in the culture supernatant, that was completely, I mean, it just took us, I mean, we didn't know what to make of it. Okay, so how is the experiment done? You take DNA and you put the culture supernatant, and then if there is nothing happening, okay, so then you will see whatever DNA you had put. However, if it is digested, the lane will go black. Okay, so that is how we know there is DNase activity. There are many different things that have been done, so let's not worry about it. The point about it is, on an agarose gel, you have DNA, you incubate it with this supernatant, and in another one you didn't put, and you just done, one is the control, the other one is, and if it is digested, so obviously we were simply not expecting that there is going to be DNase activity in a culture supernatant. So
what if it's non-specific DNase activity, like it may be hydrolysis or some other enzymes?
Possible, there could be multiple enzymes that may be there, but the surprise was that you don't expect it to be there. Okay, you don't expect to be
because the bacteria would have been also dying if the DNA was there.
So point well taken. Okay, so the point is that what is it? Because there is a small amount of the cells. Let's say you have 1 billion cells, and 1 million of the billion are lysine, and the DNA is actually the source of DNA is those cells. Okay, so because of the lysis of the cells. So to begin with, in general, what we do is we try. If you would recollect, the way E. coli would grow in culture, what is called a growth curve. Are you familiar with the growth?
Yes.
So you'll have a lag phase, and there is some exponential phase and all that. So in general, what we try to do is we pick our cultures at log phase, mid log phase, so that they are healthy, and there are, I mean, cells when they die, they also create other kinds of things to it. So what we landed up doing was that we are doing that. This experiment was originally done with that. Then we realized that this issue that you just raised, that who knows, thoda sa lysis ho gaya, and that is the reason why this is happening. So what we and obviously this is an unexpected finding. So you would also worry, and tomorrow you have to submit a thesis, you have to publish your paper, etc., etc. There you have to convince the world about. Okay, so first you have to convince yourself that this is true. So what is known in the case of Streptococcus pneumoniae is that once the culture reaches what is called stationary phase, then there is autolysis. Okay, there is a mechanism kicks in which will start the cells will start lysing, and we were obviously very careful that it's usually about if one were to use spectrophotometry, then about OD of 1 or so is when it will start doing it. So we were doing 0.4, 0.5 OD business. So then anyway, just to make sure that we are absolutely okay with it, we said okay, all right, we would make whatever that gene autolysine, which is responsible for killing, we deleted that gene from the bacteria, so that autolysis is not going to happen. Okay, so now the culture supernatant from this delta autolysin deficient strain was taken, and this DNA assay was done. Okay, DNA assay was done. We are still able to see it, which tells you the first concern that you had, or also had the same issue at that point in time. So what if there is a small fraction, whatever is there, is coming from there. So we genetically deleted the components that are responsible for the lysis, and then we did it. Okay, so this was a particular strain that we had used. Again, before you spend another 2 years to analyze, you have to be a little bit more confident. So what she ended up doing was, we said, okay, this is strain number 1. Let's see if other strains have the same thing. Okay, you would want to see, you're going to be investing time, resources, and all that, and soon enough, many of you will realize that छोटा सा experiment करना होता है, वो रहा 10,000 लग गया. Because we have experiments which are like 5 lakhs, one shot 5 lakhs. Okay, 5 lakhs is a lot of money, so you better know that this is well spent. And so she said, strain 5 more strains, did the same exercise, all of them have DNS activity, so we are now sure that we have to have. Okay, so now we can try and understand what is going on. So, but that still didn't solve our problem. We were now sure that this is happening, but the difficulty is, why would any bacteria in the world would throw out DNA? मतलब ही क्या है? मतलब काम नहीं है क्या? Right?
Sir,
Yeah.
was this DNA also recovered from Hydrophilus influenza and other strains? Like other strains also, other infections. Was that recovered in the culture?
You're saying
Reported before.
Yes.
DNA has been thrown out from other
Yes, sir.
not
It was never reported.
Not never reported. Okay, at the time she was doing this, it was not there. So we were not, as I said, you have an observation which is clearly interesting. At the same time, it makes you a little bit unsure in the sense that किसी ने बोला नहीं पता नहीं क्या है. Okay, so but since we were having multiple layers of evidence, we knew kuch to hai. Okay, I mean you should not necessarily be doubting your own data which you have generated with your own hands. Okay, so this is what happened, and then again these are different experiments. For example, DNA that she treated the culture supernatant, and what will happen when you treat
Enzyme will get denatured.
the enzyme will get denatured, and the activity was lost. So that was telling us that protein to high. Okay, so by and large, what you would know is that the enzymes are basically proteins. There are exceptions, but basically, okay, if your protein gets denatured, the activity should be lost the activity. Now that we know क्या protein है, or it's sensitive to this thing, so you can also digest the protein. So we have another protease K. आपने उसको मैं digest किया तो वो activity फिर से चला गया. Protease K per se would not have DNA activity, right? So वो चला गया. One of the other things, properties about nucleases in general is that they need divalent cations for their activity. Okay, this is true for many other protein enzymes also, but DNAs also have the same requirement. So what we ended up doing was, we said, let's take some chemical which will chelate, which will take away the divalent cations. So EDTA is one such thing which will remove the divalent cations from the solutions. So when you do that, you realize that activity is right. So this was further evidence to tell you that it is sensitive to temperature, it is a protein, it requires divalent cations, it is digesting DNA. Okay, so that is where we are, but our problem continues to be the same. Okay, so now how do you have supernatant which has more than 200 proteins being released? Which of these 200 proteins is this DNA? Right? उन 200 में से तो कोई ना कोई तो है, because all of this is telling you the story. Now, which of these is going to be there? How do you go about it? Okay, so the first aspect, as I said, I'm trying to walk you through how it panned out for Hina. Okay, so that you can connect it to what is happening in your own labs and in your whatever small project that you are having. So she initially did this 2D gels, okay, two-dimensional gel electrophoresis. Lots of spots were there, 200 plus. Now, which of those spots is causing the DNA is the issue that was there. But when the amount of the DNA, amount of protein in any given spot was very small. So to assay, to look for the DNS activity was turning into the challenge. So obviously you have to come up with some other method. So the experiment that I'll not walk you through in great detail, but the point about it was that she did a little clever thing. Okay, the clever thing was that you need more proteins to be around. So this is just this is a DNA gel. She actually put DNA in the gel. Okay, half thymus DNA. As you are pouring the gel, उसी में डाल दिया. Okay, and then she ran the gel and then incubated the gel. Okay, first the protein separated on the gel, and then there is already DNA. So when you have DNA, you can DNA separate हो गया, and she incubated the gel, and it turns out कि जहाँ DNA's activity है वहाँ clear हो जाएगा, because that DNA is lost. Okay, on a equilibrium gel, you will still everything else is bright. There is no DNA there; it will show up as a negative dark band. So it turned out that there are actually 3 bands. Okay, not one band, but she was finding 3 bands that were coming. Remember, 1 से आया या 5 से आया, हमें पता नहीं चलेगा on the assays that we did. Okay, so she said 3 bands. Now this is a gel where she has, put a bigger cone so that you can load more. So you'll notice कि ये बहुत बड़ा है. So the band corresponding to, let's say, this guy, आपने वहाँ से काट लिया. Okay, this is a gel where you can, it's like you have multiple wells, आपने उसको tape लगा दिया. So instead of putting 50 microliters, you put 250 microliters. That is the place where you have the clearing, उसका corresponding band on this big gel, it's simply, after running the gel, you cut it out. Okay, and there are ways by which you can identify what is the protein there. Okay, can anybody tell me what method is antibody? Okay, so there's a method called mass spectrometry. Okay, where you can try and identify it. Okay, so she did mass spectrometry, and then she tried to find out what is the identity of the protein. Okay, and it turns out that she found 2 proteins which are some kind of names that are there, but you go back. So these are not necessarily, I mean, how once you have the sequence, one can do some kind of analysis using various databases that are publicly available. Okay, there are homology searches that can be done. So if you know the sequence of the protein, you can do a very common base that many of us use is from NCBI. Okay, National Center for Biological or Biotechnology Information, something like that. Okay. And this particular search that is done is the acronym is called BLAST. Okay, so you put your sequence there, and it will see what are the best matches. Okay, you may not necessarily have done this, but the idea is that you can search it. I mean, YouTube is also a database, by the way. So you can put in, so you can do queries on that. So from that, it says phosphoesterases. This doesn't necessarily tell you that this is a DNA. Okay, we got some kind of hits. Okay, so now the issue was that we were trying to find out any other information that might be there elsewhere, like your question, और क्लोन को हमें पता था कि खाली ऐसा. Okay, so the closest we could do is we said, okay, from the human system, from streptococcus, of course we said E. coli, nothing matters. Sometimes there are some motifs, some sequences within the proteins which tell you this is the kinase. Okay, this is XYZ or whatever. Nothing of that kind showed up, and nothing that would remotely tell you that this is nucleus. So now we made the same thing. We have a nucleus. Nucleus activity, and from the previous gel, we were knowing it is not one. It is possible that there are three. Okay, and
last slide you have shown toward the exonucleus 3 activity is there, right?
Exonucleus activity is
exodeoxyribonuclease 3 activity is there. The second protein.
So this is based on once we figured out and all.
So that means that there is some DNA activity.
Yeah. So you have some indication, but when you go back and look at the sequence,
No confidence.
it doesn't tell you anything. Okay, when you go back and try to look at this, we said, yeah, this is conserved in all of them, this is conserved, but this has nothing to do with DNA activity. So we were back to the square one. Okay, we were back to the square one, saying that you can some progress, but we don't know what happened. Okay, then again, the thing to remember is that these kind of homologies means the level of identity between the two proteins are on the lower side, on the very lower side. Okay, so that also does not tell you this thing. There is no indication from the literature or databases that this can be a nucleus. Okay, there it is not annotated as a nucleus whatsoever. Okay, so now that we knew that there is, this is the sequence. So we went back to the gene for that particular, this is 1788. That's the SPD underscore 1788 is the name of that this gene. We went back to the gene and we cloned it. Okay, cloning and expressing it as a recombinant protein in E. coli. So essentially, you take it, you put it in some kind of a expression vector, as it is called, and you would prepare. You will. There are ways of doing it. We will not go into it, but you make a recombinant version of the protein. So if you are not working, remember the issue was we do not want to be continuously working with Strep pneumoniae, primarily because it is not the best place in the world. It's a very fancy lab. You will go, you will think, "Padani kahan ka lab hai?" Okay, extraordinarily fancy, exotic. But we don't want anybody to be there unless it is absolutely necessary. So if to make recombinant, you can of course do it in the Streptococcus, but then you have to deal with the pathogen. So we say E. coli liver. So we expressed it in E. coli. All of you are using E. coli, right? So and then we say, okay, your protein layer. Okay, and how? So we have to put a small tag to it so that will help us in purifying the protein. How many of you have used or heard about some kind of tag that you attach to a protein to help it purify? This is the second tag लगाया था. You do have certain column, nickel MPA column, and you can pull it out. Okay, so that is how she landed up pulling out the protein. So you have lots of protein. Now the issue is whether this protein is actually functionally. So if this is the real one, it should have DNA activity. Okay. So again, she did DNA and these are methods, not to worry, but basically you will end up incubating DNA with this recombinant version and it should disappear. Okay, so she was able to demonstrate that this has DNA activity, and as we had discussed previously, the enzyme has requirement for divalent cations. Which divalent cation we don't know. Okay, so what we are if you get rid of all the divalent cations, then activity is chala gaya. Now, which divalent cation do you have to add so that now the activity is restored? Fair enough? Everybody on board? So we are saying, okay, we say we try calcium, cobalt, manganese, zinc, a whole bunch of them, and try to see kisse wo restore ho jata hai. Okay, so that is the idea. I mean, different enzymes will behave differently, but you have a way of you're looking at DNA's activity. So to begin with, you have, in this case, after all of this analysis, it turned out that the activity, it doesn't have zero activity. Okay, the activity is low, but you still have a little bit of activity. When you bring in the correct divalent cation, the activity goes up. Okay, that's how this this thing works. Okay, and the other bit of it is that remember when you think about nucleases are a variety of kinds. Okay, for example, if you have a nucleus which works on RNA, what will it be called?
Nucleus.
RNA. Fair enough. You could have other versions of this, which means that you have a linear DNA, and the DNAs will cut it only from the sides. Exonucleus. You could also have another one, which is that it doesn't matter. It can cut in between as well. Okay. So we were trying to figure out what time. Okay. Now, what is the way to deal with it? We say we have plasmid DNA, UCAT. We keep it circular. Okay, no ends. We have the same thing, linearized. If you are on registered, you have linear, so that is one substrate. So we did this various kinds of stuff to find out. So now the example is that what if we don't know the activity? We are saying circular DNA, linear DNA. Circular DNA को वो काटता नहीं है, linear DNA को काटता है. What does it mean? Exonucleus, because there is no end on this. So based on this, we were able to say that it is a exonucleus. It's an exonucleus because this is we are incubating the circular DNA over a period of time, and all of them remain the same. There is no digestion. Okay, whatever you had put in the at time point 10 and 20 minutes, 30 minutes of incubation, there is no drop. So that is telling you that it is not working on it.
But sir, last time is there, that's why the protein is there and it is digested.
No, this is linear DNA. Genomic DNA, we said plasmid DNA, supercoiled DNA. Supercoiled DNA is here. Okay. So based on all of these things, we were able to figure out. I mean, the point about it is not the data per se. The important about it is how do you go about doing it. Okay, so you can have multiple this thing for nucleus A, it might be something. For nucleus B, it might be something else. What is the way to test it? Okay, that's the whole point about this entire exercise. Okay, now let's go further. So essentially, what we are saying is that there is some basal activity that was there. If you add divalent cations, you enhance the activity. And if you can cut the circular DNA, then endonuclease activity. If you don't, you do it. In this case, you cut, so it is endo. Okay. Now the issue that we were worried. So once you got the sequence, now just I'm sure you have had opportunity to think through this in some of your classes. So let's think about a mammalian cell. Okay, within the mammalian cell, you have different compartments. Nucleus. Within the nucleus, we have all the transcription, conversion of DNA to RNA, and then RNA is translated in the cytoplasm. Proteins are made, and proteins go to the cell surface. Protein goes to the mitochondria. Protein goes to other components. How is this achieved? Okay, how is this protein trafficking achieved? Signal sequence. What is signal sequence?
So, like for every protein, there are additional sequences which can be recognized. For example, ER will have a specific sequence.
So the idea here is that there is some leader sequence which will help it to target to specific compartments. That's the overall notion. Correct? Everybody on board? Okay. And if you have to target it to plasma cells, you will have another signal but of a different nature, which says, what if you have a protein which is secreted out? उसका क्या बनेगा? Obviously, it has to be targeted to the plasma membrane, but something else also has to happen कि वो बाहर चला जाए. Remember, to target exocytosis. There are multiple mechanisms by which proteins are secreted. Okay, so first of all, you have to target it to this thing, and two things happening. One is it gets targeted to the membrane, stays there. The other one is gets targeted to the another protein that goes to the membrane and it's released out. So there is a distinction there. Anyway, so we.
I have one question.
Bata.
If there is a toxin which can only be secreted out when it is interacting with the host, like if the host is not there, then the toxin does not get secreted out. So, like,
a whole bunch of typically something which is in this case something which is destined to be there. If the host cell is not using it, it doesn't. I mean, some of this is in response to presence of this thing. It may be if it is not there, it may not necessarily be there. So these are all typically most of these are released by default. Okay, but then for any, I mean, in general, there are mechanisms by which they can, whether this particular hypothetical toxin is inducible, meaning that in response to a certain cue from the host, okay.
Signaling pathways, similar to the neurotransmitter release.
So, as I said, you can have a whole diversity of situations where this can happen. It may or may not necessarily be that because it is now there, the presence of host is sensed by the bacteria and therefore it starts releasing. Okay, one can have situations where this might also happen, but there's a whole spread of it. Okay, so now what we learned was that there has to be some kind of a signal peptide that would help you get released. So now when we look back at the sequence, there is no signal peptide. We did a whole bunch of other analyses. We said relax the condition, kahin se to kuch aata hai, kuch nahi aata. Okay, now we have just summarizing what we have at this stage. We have a protein which we are convinced that it has DNase activity, recombinant protein बनाया, उसका भी activity है, but वो release हो कैसे ना इसके लिए कुछ समझ नहीं आया. No signal peptide. Okay, we will say okay because it has to be targeted to the membrane. Maybe it needs a membrane को target करना तो hydrophobic set of amino acids. एक भी नहीं.
So sir, if you pair it with some master like PFP or something, of course it is very difficult in bacteria.
I mean, one could try and try. If one remember our focus was to try and find out what are the proteins that are responsible for pathogenesis. Okay, I mean these are absolutely interesting aspects of पहुंचा कैसे. You can make it a question and then chase it. Right?
Maybe you can synthesize it. Like you can reverse the process. Like we are on the protein sequence. You can make a synthetic oligonucleotide and can be used to sort like PCR amplification, and you can amplify the specific genome of the bacteria, and then you can see. Then after sequencing that area, you can see if any leader sequence is there.
We did all of that. What you're saying, we did all of that. We looked at the genome. In fact, the complete genome sequence of that humanase is available now. Okay, so we went through everything, but it's just not there. Okay, but again, obviously, remember this is an observation which nobody else has, and therefore when you tell people, they will say, "यार दोस्तों गड़बड़ है." ऐसा होता नहीं है, books भी नहीं कहती. Okay, so now what do you do? Okay, and then इसमें signal तो मान लीजिए it is very easy to assume यार तो कुछ गड़बड़ है, इसमें तो ये है नहीं, peptide ही नहीं है. Now while at this point in time we were trying to look for कि हाँ भाई it is happening. We have seen 6 strains and we are noticing this happen in all of them. So it is in our mind it was real. Okay, so then at that point in time, Hina came across a set of papers which was saying that bacteria can make small vesicles, nanometer scale vesicles as a part of their normal physiology. Okay, so with that kind of set us thinking, यार इसमें से भी कुछ तो नहीं हो सकता. So you say, okay, whether this enzyme can be packaged into this small extracellular vesicles and they are anyways being released. तो आप जब सुपरनेटेड में ढूंढने लगोगे, उसमें आपको नैनो स्केल में तो आ ही जाएगा. It's almost like in solution kind of. Okay, so in your supernatant it shows up, and we are seeing activity. Now, how in the world do you? And if this is a electron micrographs, these are the ones which are budding up. These are being released from this. So the next thing was a picture. Once we thought maybe let's test this out. Okay, what if that is well? So the principle of is there's this budding happening, and contents from within the cells, cytoplasm can get packaged, and then they are released Okay, and what has been observed is that you can have DNA in this. Okay, I mean this is not capable of replication on its own. Okay, but just because ये budding हो रहा है इसमें जो भी उस time पे समान था वो इसमें आ गया. And now this is extracellular vesicle. There are components of the membrane, and as we learned a little bit more, then we realized that the composition of things up here versus what is there in the whole plasma are not necessarily the same. There are differences that are there. You can tell them apart. Okay, so this was the thing. Our point was whether this enzyme could be our DNA. Okay, so now let's go back and try and understand a little bit another phenomenology before we come back to our story. So here what has been depicted is a neutrophil. Remember, we said for Streptococcus pneumoniae, the main productive process is antibody mediated. Okay, but remember, finally some phagocyte has to take it up and it has to be digested. Now, when a bacteria, Streptococcus pneumoniae, remember we are using it as a model. This applies to a whole bunch of other bacteria. Okay, just remember this. Now, when it comes in contact with the host cell, it is going to, let's say, neutrophil. Typically, it is going to get taken up, and during the time we were looking at this, there were some notions that were coming up that neutrophils release, their chromatin. Okay, so this is an updated version of the understanding at that point in time. ऐसा कुछ होता है करके, मतलब somebody observed that neutrophil से chromatin release हो गया. Okay, this is an updated 2021 version. There, you have bacteria can induce the formation of what is called neutrophil extracellular traps. Okay, this is the next that is there in the title. Okay, and these components have histone hai, chromatin hai, so histone obviously is expected. You have LL37, which is an antimicrobial peptide. These are representative. There are tons of it. Okay, neutrophil has lot of these granules that are there. वो इसमें bomb की तरह तो पड़ा है. You have to just throw and that's it. So, and then you have various other enzymes, myeloperoxidase, neutrophil elastase. These kill the bacteria. Okay, so time up हो गया.
I think any is non-ethnicity or
Endonuclease.
Which any
any the
neutrophil elastase. Okay,
So I just have a question because the bacteria have this immune system where they can produce restriction endonucleases, right? So how different it would be from the
so that is for stuff that is getting into the cells. Okay, so let's say there's a virus
they're
bacterial
not
phage.
secreted out,
Yeah,
but
okay.
yeah.
So that is if there is a virus bacterial phage attacks, which is going to go into the system, and then that mechanism kicks in at that point in time. Okay, so now what we have is that these are just representative. It is loaded with all kinds of antimicrobial activity, peptides as well as enzymes. So, and this is a lot more detailed than what we had. At that point in time, there was an observation saying that कुछ chromatin निकलता है. So we were trying to again remember we were trying to piece together कि हो क्या रहा है, because we have some very interesting leads, but at the same time we were not sure कि how does it all gel. Okay. So we said all right. We said neutrophils. Now,
so this is also latest finding, right?
That-
Your research was done in March earlier.
Yeah, so
that's
now
why it is really.
no, that's I'm seen. I can just flash the data and do it.
Right.
But for this kind of audience, लैब में वो रोज चीज होगा ना, तो बोले यार सर ये आज आया नहीं, आज ये आ गया, ये आया और तुमने मिस कर दिया कि क्या हुआ, होता है ये तो ठीक नहीं है, तो लिटरेचर से मैच नहीं करता, साइड में कर दो. ठीक है, most of the time that is what is going to happen. Okay.
Students only wants the golden particle.
Yeah. So I'm just saying that you have it, you believe what you have.
Right.
Okay, remember this. This is not Dr. Sehgal speaking. I'm talking on behalf of the student. Okay, so that you can identify with the student. Okay, उसके साथ क्या बीत रही थी? Okay, that was the whole point about this exercise. This is what she was living through. As a part of it, you guys वो excited है, one day you come, sir आज party करना है, बहुत अच्छा result आ गया. And next day sir पता नहीं क्या होने वाला है. हाँ, मुझे नहीं पता सर, इसका पता नहीं, कुछ नहीं हो रहा है, I don't know what to do. So this is real thing, okay? So I mean all that was there is ये तो आज ये 2021 है, at that point in time there was some neutrophil से DNA chromatin निकलता है, that is where we were. Now I mean literature doesn't connect any of that, okay? So therefore you don't know. So anyway, so this is just again this is a more recent thing. Just to tell you that neutrophils, there are two processes that are there. One process is what is called slow death thing, which is called, this is a combination of cell death and which involves release of nets. So this becomes netosis. And in this case, the neutrophil dies, okay, by a process called apoptosis. You may have heard of it, but take it as cell death. And these nets are released. Okay, basically you have the cell plasma cell membrane has ruptured, and there is a release of nets. The other one is a little bit different story. Okay, the neutrophil doesn't die at this point in time. It will suddenly release a lot of nets, but the cell will be alive. As far as the bacteria, so this is pretty much, you have a subset of all the chromatin that was there, but for functioning purposes, बाकी सामान इसके पास है, so it is alive. So it's like alive. You can show activities of various kinds. So it is called cytoplasm. They're not even using the term cell. Okay, but anyways, you have this nuclear chromatin is all, there is degranulation. There's all that all kinds of goodies as far as the host is concerned are all put on top of this. So you have the nets around. Okay, so this is so now we again. How do you test whether these small vesicles are released or not? See, literature says release होता है. आप करके देखिए कि release हो रहा है कि नहीं. Okay, you can sit textbook से ऐसा कर लो, वैसा कर लो, good enough. Here it is story as on the bench. Okay, so she made a bigger culture, 1 meter culture. Okay, remember this is a BSL-3 biosafety level 3 experiment. So हाँ, तो वो आप उसको बिल्कुल संभालना अलग story है. Okay, so not to say कि this can't be done. Those facilities are just top of the line, and I won't say zero risk, but if you are careful, you will do perfectly all right. Okay, so culture लिया उसको, you have to spin it down, do some kind of density gradients, and at set particular, because it will depend on the density of the vesicles that are being produced, and they are taken out, expirated. You take it to electron microscope. Now the vesicle है भी नहीं है, because you pulled out something from a tube. Okay, according to literature, where some other people have done, if you remember plasma, if you just do fractionation, I'm sure all of you have read this as a part of your cell biology or something like that. You can break open the cell. You say इसको इतने spin करेंगे nucleus आएगा. You spin it harder with this, you will get something else. Various components that are a part of this. So we took made use of it, and we say ये वाला electron microscopy. We said हाँ ठीक है, with low small vesicles are all over the place. You can measure it and all that. He said हाँ ये तो हो गया. See, remember this is not all that easy. वहाँ पे आप electron microscopy sample तैयार करिए, वहाँ measure करिए, जब आपकी बारी आएगी आप लोगे and all of that. ठीक है, so this is it's not हाँ भी अभी देखा कल हो गया. Okay, so anyway, so once that is done, she said वो fraction लिया, and if this indeed had DNase activity, we should be able to take that fraction and do our old-fashioned DNase activity. ठीक है, वो हमने उसको करके डाला तो उसमें हम लोग डाइजेशन हो गया. Okay, so what it is telling us is that DNA has activities associated with this fraction. Okay, again I will not walk you through this. We have all the gels, etc., and we have done it. But DNase activity, we said do it with another assay. Okay, which says if there is something peculiar that is happening, because remember, biology is very diverse. There are so many mechanisms that are involved, and it's full of surprises all the time. Okay, and the processes are a lot more complex than some of the ones that we study in other subjects. Physics, chemistry, and all those are rather complicated. So anyways, we did another assay, picogreen assay. Okay, so this is another way of looking for DNase activity. So this is just what I have just mentioned previously, that so now we were beginning to, we made a hypothesis that maybe is there a connection between this EVs and this business of net formation. कि हाँ भाई कुछ इनका आपस में कनेक्शन है कि नहीं. I remember these are nascent ideas. आप you discuss with the student over tea. उधर देखते हैं करके देखते हैं इसके आता है. Let's see what happens. Okay, so when you take that, then you have no worry about how do you execute it. Implement that experiment in the lab. आप उसको सोचिए. Who knows it is there or it is not there. Okay, obviously it has to be tested. So the funda was that hypothesis ये निकल के आई कि हाँ भाई what if आपका बैक्टीरिया है, होस्ट में इन्वेट कर दिया, न्यूट्रोफिल के साथ कांटेक्ट में आया, either the neutrophil took it up and it killed. The other one is neutrophil, because of some other things which we don't understand, it is releasing nets, and it is like taking the nets and throwing it on a wild animal. And उसको आपने पकड़ लिया, कैप्चर करके उठा ला. Here on the nets you have all this antimicrobial activity of 1000 kind, and those are going to kill the bacteria. Now obviously bacteria बोलता है आपका बेटा मैं तो ऐसे फंसने वाला नहीं, ठीक है? So what if the bacteria is throwing these nucleases to get rid of the DNA component of this and escape the food beta? Okay, and then go to other parts of the body. This was the notion at 4th year of study. ये कि हाँ भाई ये हुआ कैसे करेंगे?
Can we radiolabel the DNA into nucleases? We can give them and
Okay.
15 million.
Okay, all right. So we'll look at that. Okay, how to test it. Okay, anybody? I mean, as I said, the whole point about was not. The point is help you think through how you would do it in real life. Okay, some version of this you will face in whatever projects you are doing. And if you understand, अभी ऐसा नहीं कि हमें आराम से बैठ टहलते हुए चलेगा और फटाक से हो गया, ऐसे कुछ नहीं होता. Okay, you might have something else. It may be a chemistry lab for all you know. Situation is not going to be because you are in the unknown territory.
Right.
If you already knew it, ये तो करने का फायदा नहीं. MSc करिए, आप PhD करते तो it's a different degree. You are in the territory where nobody might have an answer. Okay, so that is
And
the
no one
point.
knows how to do it. Right?
Yeah,
That's
that's
how you
what-
are here.
Yeah, that sounds. So you have to figure out how this could be addressed. That is the point. And as I said, ये करो ऐसे कोई लिखा हुआ नहीं है. आप वहाँ बैठेंगे, आप सोचेंगे और फिर एक्सपेरिमेंट डिजाइन करेंगे, फिर उसको टेस्ट करेंगे. So now you can think about 15 experiments, see what is the most reasonable, most fast. इसके आपके पास रिसोर्सेज हैं, आपके पास पैसे हैं, आपके पास इंस्ट्रूमेंट है, वो उससे आप कैसे एड्रेस करेंगे? There is no one way. Please.
Sir, I have an alternate hypothesis.
बताइए.
Sir, like जरूरी नहीं है कि सर वो नेक्स्ट को एस्केप करने के लिए होगा. सर मे बी सर इट इज बैक्टीरियल इवोल्यूशन, क्योंकि सर लाइक हमें पता है कि बैक्टीरिया ट्रांसफॉर्म होते हैं, एंड वेल नोन की स्ट्रेप्टोकोकस निमोनिया भी ट्रांसफॉर्म होता है. एंड लाइक सर व्हाट इफ बैक्टीरिया ये सोच रहा हो कि मेरी वायरुलेंस खराब ना हो जाए, किसी मतलब फॉरेन जीन अगर आया वो उसके साथ मतलब इनकैप्सुलेट हुआ तो मतलब देयर इज अ चांस है कि उसकी वायरुलेंस खत्म हो सकती है. सो दैट माइट बी द रीजन कि वो पहले ही अपनी एनवायरनमेंट में एक्सोन्यूक्लियस सिक्रीट करके रखता है ताकि कोई फॉरेन एलियन डीएनए होगा दैट इज डिग्रेडेड, एंड सर इट विल इवेंचुअली स्टॉप द ट्रांसफॉर्मेशन एंड उसकी वायरुलेंस कंटिन्यू रहेगी और दैट इट कैन इन्फेक्ट पोस्ट मल्टीपल.
So the hypothesis here is that ये नेट वेट का कोई चक्कर नहीं. Okay. So the original purpose, the real purpose of the physiological function of the secreted DNAs is to get rid of foreign DNA. Okay, that can which was preventing to get into the bacteria, because if you get foreign DNA, it can get integrated. It can negatively impact the resilience properties of the bacteria. So let's try and see how we can look at this. Okay.
And sir, is there any specific pathway which is now described, like why nets are produced, sir?
Why nets are produced?
Why nets are produced?
So, like, if the DNA is going inside the vacuole, that means it is being like to be degraded, right? Like vacuoles are used for waste.
So even
storage
this
of
DNA, even it's produced outside, like like it's just bombarded out. Like, it's fermented. It can be bombarded directly outside, or sir, it is in the vesicle. I don't think, sir, it would be in the
अभी हम लोग अभी at this stage we don't know.
It is a
He not
regulated
did not know.
mechanism, right? It's not like something
License
abrupt.
Exactly.
So at this point in time, we had no clue. Okay.
Okay, sir. Why that is actually no clue, sir.
No clue.
Mechanism is not
No
known.
clue. ऐसे उस टाइम पे कहीं some paper says that यहाँ पे imported degrees. Okay. We'll come to your funda. We say इसका हमारे को inlet से कोई connection नहीं है. It's the bacteria is producing it for something else. And the something else is that it doesn't want to get transformed unnecessarily. तो आप जो बाहर DNA है और मिल्यू में और भी चीजें हो सकती हैं, उसका DNA आपके अंदर आके वो हो सकता है अच्छा हो और मोस्ट ऑफ द टाइम गड़बड़ करेगा तो यार सारी डिस्टर्ब. ठीक है, possible. ठीक है, possible. Okay, so I'm not negating that, it is possible. Okay, now the context in which we are वो बाकी the one has to remember this that streptococcus doesn't grow out there. Okay, if you take out streptococcus and you put it in a tube, even if you put it on ice, it will still die.
But you said like it's there in our throat. So then along with streptococcus pneumoniae, there are two other bacteria also.
I was not discounting it in any way. Okay, I was not saying that he only is it. Okay, I'm just saying that in the real world, this pathogen is incapable of. It's not a free living pathogen. Okay, so it is dependent on the host heavily. If I have to culture this bacteria in a chemically defined medium, which means you know exactly each component, it takes 60 different components to grow it. It cannot. It needs all 20 amino acids. It needs all nucleotides. Okay, compared to any typical bacteria, the amount of genome is half. Almost everything जो machinery होता है ना, biosynthetic machinery, वो सब out है, dropped from the genome. Okay, so its ability to survive outside is very low. Okay, so it pretty much has to be in some host for its propagation, not like the virus situation by and large. So that is why we were favoring this situation कि यहाँ पे host में survive करने के लिए आपको बड़े पापड़ बेलने पड़ते हैं. ठीक है, उनमें से एक पापड़ ये है. Right? So I'm not saying that the DNA as a genetic material was discovered in the same strain that is being used in this experiment. Same strain. Okay, the original discovery that DNA is a genetic material. Remember this. It's the same D39 strain that is being used in the study. Okay, so transformation होता है और पहली बार anybody figured it out was the same thing. Textbook में यही strain है. Okay, so the point about this funda was that कि हाँ भाई, what if there is a connection? Okay, so how do you go about it? And as was pointed out, we have to have some way to label it. You were saying radio label, but some other ways can also be used. And like you don't want anybody to be going into the BSL3 lab, you don't want people to be using radio. So हम लोग बोलते हैं कुछ radio label के बिना काम हो सकता है, उसी से करिए. We don't want students came here to learn, not to get injured by various ways. Okay, so how do you label it? Remember on those chromatin, you had histones, you had antimicrobial peptide, you had neutrophil elastase, myeloperoxidase. Okay, तो हमने एक एंटीबॉडी वो उसके अगेंस्ट ले ली. Okay, वो जो डीएनए के ऊपर these proteins that are there, so I think वो myeloperoxidase. Okay, so these are human neutrophils, blood. इन्होंने बाकी लोगों के लैब का सारे बच्चे डोनर्स of the experiment.
Right.
खून पिए.
हिंदी में हम बोलते हैं खून चूस लिया.
आप खून चूस लिया. हाँ, वो भी खून चूस लिया. ठीक है, एक बार चला एक समोसा खिलाया और 5 ml ले लिया.
Malaria is very common.
I know. वो 5 ml ले लिया. ठीक है, so people still whenever she comes from Germany, तो उससे तो तुम एक केजी का चॉकलेट तो ले ही लेते हैं कि वो you are there because of us. 5 ml करके गया है. ठीक है, so the point about it is the human blood neutrophils. Now for the experimental purposes you can use a chemical PMA, okay, which will induce the neutrophils to to release these nets. So those were released. I will not walk through the entire thing. The point about it is, that you can induce this, and how the experiment is the following. You take in one case you have not added EMA, so there is no neutrophil extracellular traps. The second one you have added, so that's experiment 2. Third one you have put the wild type strain. Okay, because remember it is supposed to have antigenous activity. You made a mutant which lacks this gene. The activity is supposed to come down. Okay, so आपने जब वैसे किया, then you realize that wild type ने तो वो clear कर दिया. Next, the mutant is not able to do it. Okay, it is highly compromised in that. Okay, so that tells you I have this seems to work and seems to explain. So these are data downstairs. Okay, so what we are saying is, you can, the other experiments that have gone into it, let's work and see. So these were done in multiple ways. आपने वो secretome from the wild type, secretome from mutant से डाला, तो एक ने इसी slide पे these are confocal microscopic images. They can do even better than the whole bacteria because it's concentrated. Then third experiment, you do it with recombinant protein, and as a control you use some outside protein which is that does not have DNA that should display. So we know there is a connection between the mutant and the secretome and the recombinant protein. ये सारी चीजें उसको degrade करते हैं, so that was beginning to give us confidence that how it would work. Okay. So obviously the recombinant protein, now it is just that enzyme, it's going to work much more efficiently than the whole bacteria which is releasing. How much it is releasing we don't know.
The concentration is
very high. So it does lot more efficiently. Okay. Now, while this was happening, another group landed up. They were trying to understand what you were just mentioning, kind of DNA uptake. Yes. From transformation, remember, transformation was originally discovered in the same strain. Okay, not for this strain globally, first time. So those people were trying to understand how this DNA is taken up. What is the mechanism of uptake? Wo DNA जब आता है तो ऐसा नहीं कि it simply walks in, no? So they were trying to do that, and in the process, they came to this situation which they were saying that DNA, I mean, that hypothesis not very well worked out, but the idea was that you have DNAs and maybe उसका एक strand काट देता है और एक strand cell के अंदर जाता है, तो एक membrane bound nucleus, endonucleus A नाम दिया उन्होंने, वो उनको दिखा, तो which they said that this is the one which is helping in the transformation when the DNA, like free floating DNA comes in, it is one strand is cleaved and then the one strand goes in, and उसके अंदर जाके वो अंदर जो single strand is more likely to exchange this, that, and the other. That was the standard. So now at this point in time, we said DNA release कर रहा है, हमारा वो लग रहा है vesicle में packed hypothesis. We were able to see that the vesicle has DNA activity, and it is released, and then this new player comes in. And remember, the reason it is important to know कि ऐसा नहीं कि तुम अकेले मुन्नी और मुन्ना अकेला ही चले जा रहा है, वो बहुत सारी मुन्नियाँ और मुन्ने वर्ल्ड वाइड घूम रहे हैं, ऐसे स्पेन में भी बैठे हुए हैं, यूएस में भी बैठे हैं, वो लोग भी कर रहे हैं, वो भी अपना दिमाग लगा रहे हैं, तो उनको वो कहीं कंप्लीटली डिफरेंट चीज कर रहे हैं, उनको न्यूट्रोफिल में, और हम आए विरुलेंस फैक्टर से, बोले यार वो 4 लोग को मेम्ब्रेन पे कर रहे हैं, सिक्योरिटी पे कर रहे हैं, लॉजिकल है, कोई ऐसी बात नहीं, उसके कर लेते, ठीक है, और वो वहाँ वो बोल रहे हैं डीएनए कैसे जाता है, उसको कैसे एफिशिएंट किया जाए, ये किया जाए, वो किया जाए, वो एक आ गया। तो वी हैव नाउ कि अब ये एक नया, इट्स लाइक मूवी में एक विलन और देख मूवी देखते हो, हाँ वही वाला चक्कर, तो वो बीच में टपक पड़ेगा। So now we are saying कि हाँ भाई, tomorrow you have to convince yourself, your examiners, and also paper भी भेजोगे यार। So what will happen to those? वो बोलेंगे यार ये तो यार वो उसकी वजह से था, इसकी वजह से था। So now we have to deal with not only what we had but also explain what is happening in the other case. So we said ठीक है, हाँ DNA activity तो उन्होंने उनको वो एंडोन्यूक्लियस बताया ही नहीं है, तो उसमें है ही है। So what was done is we said, okay, we deleted. We already had the mutant, wild type mutant with 1788 missing, and we created another mutant which was there and a mutant and the double mutant. Okay, यहाँ पे individual contribution to this entire process पता चल जाए. Okay, so what we noticed was that यहाँ पे if you I don't know, this is online people may not be able to see. This is the end nucleus. So ये wild type में इतना degradation है, इसमें थोड़ा सा कम होता है. हमारे वाला डालने से ये इतना कम हो जाता है. This is okay. Anyways, so this is how much you will degrade because whatever all the endonucleases है सारा मिला के इतना है. जब हमारा वाला गया तो यहाँ से इधर आ गया 40% पे पहुँच गया. ये endonucleus करता है. It goes to drops a little bit. Karta, but little bit. When you drop both of them, it goes dead. So now, again, this is all data from all the confocal imaging. So we'll come to this thing. So both of them are contributing, but our protein that we have is contributing much more than the other. So we said, overall, if this is real and it makes a difference, then we should be able to see पैथोजेनेसिस में देख पाएं और बाकी चीजों में देख पाएं हमें रियल एनिमल में क्या हो रहा है. So
that is membrane bound, right?
This one is membrane bound. This is secret. Okay, so Hina infected animals again all keep the experiments, and the most infectious part of the entire exercise is the animal experiments because they are becoming even more and more potent. So we infect the animals intraperitoneally. So this is sepsis model. It goes to all the organs, okay? And she pulled out the spleen, blood, and the lung. Remember, lung is because pneumonia, and it also causes sepsis. So blood में भी आ जाएगा, and spleen is where all the action is as an immune system. So what she finds is that in the case where you have when you get rid of the this mutant के साथ करते हैं, because your loads are much lesser, okay? Because it's not able to clear the nets, and therefore so what we found was that there is a correlation between the amount of the bacterial load in all these organs versus and that phenotype correlates with the presence or absence of this mutant. Okay, this is then this we looked at lung pathology. So I mean, we are not the right people to look at lung pathology. So we requested ऑल इंडिया इंस्टीट्यूट के डॉक्टर, उनको हमने बताया कि हमारे पास सैंपल में क्या क्या है. उनको बोला यार ये 20 स्लाइड है, आप इसको देखिए और पैथोलॉजी के ये 5 मार्कर्स हैं, इन्फ्लेमेशन, इन्फिल्ट्रेशन, ब्ला ब्ला ब्ला, और आप फिर उसके बाद बताइए. एंड सो व्हाट कम्स आउट ऑफ इट इज दैट in the right in the corner you have the wild type, which obviously is causing the pathology, which is more severe. That histology score is on the y-axis, and with the mutant the pathology is reduced. Okay, the pathology is reduced, and this also comes up in another kind of experiment on the top there. This is a mouse survival experiment. Okay, so how does this happen? So you have the bacteria, you infect mice with the wild type bacteria and another group of mice with the mutant bacteria, and you go every 12 hours or 8 hours and see how the animals are doing. The symptoms that are developing, you store all the various symptoms, and from there you will be able to tell the severity of the disease. And we don't allow because there is ethics involved, you don't allow the animal to die, but you know that ये इतनी दूर तक पहुंच गया, वो ICU पे चढ़ गया, उसके बाद वेंटिलेटर पे चढ़ गया, तो अभी चढ़ गया, ठीक है, तो उस टाइम पे you euthanize. Okay, so basically that is, so based on that what you find is that on the mutant, the survival of the mutant in this right hand side bit. So again what we find is there is again a correlation between what it also shows up in the mouse survival assay. Okay, so that is what was giving us the funda that if the whole, I think we have a model or something like that. This is what is happening. It's just a summary that how they want, the mutant is deficient strain is attenuated in virulence. Okay, it has lower bacterial load in all of those. Okay, different organs, lungs, blood, and spleen. And in the mouse sepsis model, this was also confirmed in the pneumonia model. You have it shows attenuated virulence. Okay, so that was helping us figure it out. Now we're trying to put things together. So now remember, here we have Streptococcus pneumoniae, and we have end A, which is membrane bound. And one of the proteins that we found was that this activity is associated with the EVs. So now the very clever. Okay, in case the bacteria comes in contact with the host cell, so आपके पास endonuclear is one, you can act, just it is right in proximity. And for the ones where the DNA is a little further away, it's like mothership, who have small ship, load it with the ammunition, and say go and attack. So this is acting at as a distance. So overall, what is coming out of this entire business is that it obviously it's a novel, extracellular vesicle associated, endonuclease, endoribonuclease, which is facilitating the escape of the bacteria from neutrophil extracellular traps. And if you get rid of this, your virulence is compromised, therefore it is essential for full virulence. Okay, so as I said, this work is done by these two students in the lab. The point about it is that along this journey, you had very many ups and downs and excitements, and we are all along. Okay, and this is not radically different from any of the studies that you would be, your heart is there in the project. It goes over. Okay, and also to tell you that it's not that you are working in isolation. And you have to deal with it as which you obviously didn't anticipate. You land up saying, "Yeah, ये तो possibility हो सकती है, आपने try कर ली," and then you know that experiment has no problem, the controls are okay, and you didn't, it is, you are sure this is certainly you can roll this out. ये नहीं हो रहा. Okay, जब तुम्हारे पास 3 options है, तीनों rule out हो गए तो क्या? So one may have to go back and look at literature, think out of the box, and it is not necessarily कि वो किसी बाहर नहीं किया तो उनका जरूरी है कि अच्छा हो. Okay, it does not necessarily this thing. So you might absolutely have a situation कि यार तुमने experiment किया, तुम देख रहे हो, तुमने उसको एक और additional way में आपने उसको test करके देख लिया, it holds up, you better believe what you have. वो जो लिखा तो लिखा और लिखे रहने. You would counter them saying कि वो लोग का ठीक नहीं है, और and you can have new players in the game. Okay, so this is Hina's story, not my story. So just wanted to tell it from her standpoint rather than giving a. I mean, I can flash this off and it will be quick and done. So if there are any questions, I will be able to have. I'm happy to answer, and I hope that.
Can I have 1 minute?
Yeah, around it.
He's one of the most interactive person probably, and he's working with a friend Italian, so you can understand the immunology.
Bichara.
Sir,
We can. I mean, this is not necessarily restricted to this presentation. See, the whole point about this entire program is that you can understand how it happens in real world.
right.
Okay, and how it works, how journeys are made. Okay, so that's basically the point. So it is not. It doesn't even have to be anything connected with this. I am happy to answer questions.
I have a hypothesis to propose.
Yeah.
Like, I might sound childish, but sorry if I do so. Like, I was studying about bacillus anthrax. So like, I was studying about the capsules in a broad way, sir. Sir, I have encountered the encapsulated strains of bacteria. And sir, when I studied these strains, so one thing that was common in all these, that this that this capsule prevents the endocytosis by the macrophages and neutrophils. And even sir, it prevents the bacteria to get attacked from the lysosomal enzymes. And why so sir? I had followed Bacillus anthrax. So basically what I had seen in Bacillus anthrax is the outermost capsule layer was made up of poly-D-glutamic acid. Point to be noted, sir, poly-D-glutamic acid. And the most of naturally occurring amino acids are L-amino acids. And sir, these are unusual amino acids like poly-D-glutamic acid. And sir, but if sir, we can like they can be an isolated target for a bacteria. Like sir, we don't have any like D amino acids in our body, so that we can. So I have identified a unique target, sir. So, but if sir, if there is any way, so we can remove this capsule, and we have identified the like it is unique, sir, to this. And sir, like after that, even our normal immune response can even eliminate out all these pathogens. And sir, like can we make a similar approach in other pathogenic bacteria, like say Streptococcus pneumoniae, if we identify that something unusual, and some unusual things are there, sir. So like, okay, I want to explore the area.
So let me just expand. You're right in saying that. See, remember, we are talking about an extracellular bacteria.
Yes, yes.
A bacteria which is capable of replication outside the host cell, not necessarily outside the host. Okay, if it is in blood, it is not within RBC or macrophages. It's outside the blood. It's not inside. Now, for just for comparison purposes, let's say it might you may be able to relate to it better in NAT's lab. If it's a bacteria, tuberculosis, salmonella, these can get into the host cell macrophages. For example, वहाँ वो उनके लिए मजे होगी.
Yes.
ठीक है. अब एक बार जाने के बाद सारा खाना वाना सब बन के आ रहा टेबल पे और बाहर फोर्ट बना हुआ है. Where are you protect like?
Yes.
BC. आप वो कोई नहीं आ सकता. आप सब बंद है उसके अंदर सब एक सांस में आ रहा है. Okay, so the point I'm making is, so for this kind of a pathogen, we'll come to your deep question. वो if it goes in by anything, तब तो हो गया. It has no means of यहाँ पे आपने lysozyme के साथ fusion बनाने के लिए रोक लिया. You have changed the machinery within the host in a manner that now the host is working in your favor.
So the MTP does so.
No, that's what I'm saying. That's why I specifically raised because it's a TB lab. No? So वो उल्टा करता है. For the extracellular bacteria, if it goes in, वो तो गया. There is absolutely no way it can
So the
survive.
capsules prevented. I have studied.
No, fair enough. So I'm saying, what is the mechanism? I mean, these pathogens, TB, Streptococcus, have been there for for ages.
Yes, sir.
So, now what will an extracellular bacteria do to prevent going inside the host? If it goes, the end. So you don't go in.
But
The capsule is not going to allow it to go in. Okay, because of a variety of things. You will come up with or various mechanisms which are a little unusual. Use of three amino acids for various. There's a pentapeptide which is there. Some of this also applies to Streptococcus as well.
Yes, sir.
So it doesn't allow you to go. Now, I'm just to give you just an example to understand the importance of this capsule as far as from the bacteria pathogen point of view. Streptococcus pneumoniae, all clinical isolates have the capsule. All, no exception whatsoever.
Yes.
Okay, number one. Second thing is, if you get rid of, and for example, we have strains in our collection. 5 CFU, 5 bacteria of Streptococcus of that particular strain. If you inject into the mouse stands no chance of surviving. 0 chance. It is deadly. Okay, itna lethal hai. Okay, we were not able to get it to 1 because counting becomes an issue. Okay, if you get rid of experimentally, you can get rid of the capsule.
Yes, sir.
We have a strain where we have got rid of the capsule. No capsule. You can put 10 to the power 9 in the same mouse, nothing happens.
Yes, sir.
10 to the power 9 versus 5. Just not 5 into anything, just plain number 5.
Yes,
डालने से वो मर जाता है.
Yes, sir.
If you get rid of the capsule, 10 to the power 9, nothing happens. Okay, it is clear as if to चने खा गए और पता हो गई बात. Okay, so that is the important, that इतना उसका role है.
Yes, sir.
ठीक है. Now one more experiment. You say I have the wild type, and you have the mutant which lacks the capsule. Transcriptome analysis. Okay, you will know कि कुछ आगे पीछे हुआ है कि नहीं. If you look at the transcriptome, you will think ये दोनों अलग-अलग species के हैं. इतना फर्क हो जाता है. Your motion के can we do something to, get rid of the capsule?
Yes,
okay, I'll also come to that in a little bit. So as soon as you do that, it is so hardwired in the system and integrated with all the entire metabolism within that, तो पूरा का पूरा ही पलट जाता है. तो बोला 4 जीन्स ऊपर हो गए 20 नीचे हो गया, ऐसा नहीं है. It is so different. It is so different. You might have a better match with another organism than within this two. इतना फर्क हो जाता है. तो वो इतनी जोर से मतलब now just to add a little bit on the streptococcus side of story. We also explored this the notion here.
Yes.
One is you say okay हमने virus vaccine लेके करते हैं. Vaccine तो चल ही रहा. I mean the reason why we are on kind of past for most part of the vaccine of this pandemic is courtesy immunology. Otherwise आप इसमें से कुछ तो crash करा देता है. ठीक है. Essentially vaccine. The other one is आप उसमें drugs बनाइए. This is a living example where the vaccine has completely 99% versus 1% contribution by the drug industry. For getting rid of this is a real living example. तुम्हारे सामने की बात है. So that is what it. So we said क्या देखो एक तो आप vaccine बना लीजिए. Our idea of capsule what happens in step pneumonia example. There are 100 different capsules that are there. 100. Okay, so आप एक के अगेंस्ट बनाएंगे तो तब तक वो नया स्ट्रेन आ जाएगा. ओके, उसके साथ वो कैप्सूल इतना फर्क है कि वो उसके साथ वो लगेगा कि नया बीमारी के साथ आएगा. ठीक है. So what we thought was we need to target something which is central to this, which is polysaccharide. There are repeating units in there. Okay, 100 different serotypes. And the repeating unit can vary from 3 to 8 different monosaccharides, different kind of linkages. So each of them is a different serotype. Serologically, it is handled by the immune system as a different strain. आपने you get the serotype 1 से आप infect हुए, तो जब अगली बार देखोगे serotype 1, then you have memory cells. You will remember that you had this infection.
Yes.
You will come back with much more force, and your antibodies, for example, will come with much faster kinetics.
Yes.
Secondary response. लेकिन serotype 2 हो जाएगा ना, वो उसके वो ऐसे handle as if primary है.
Primary.
And there are 100 of them. So we were saying कि हाँ ये पाथ पूरे जो इनकी synthesis, each of these serotypes have at least one gene. 10 से 15 genes are involved in the synthesis of the capsule. At least one gene which is unique to that particular bacteria, that strain. If you take that strain and you do an NCBI search, nothing comes up. It is so unique कि वो आया कहाँ से आपको पता नहीं. And there are hundreds of genes which are completely unique to that. Each of the strains have at least one gene to synthesis में, और जिसके लिए कोई मैच नहीं. कोई मैच नहीं है. I mean, all of us are related in some way or the other. It goes up blank. ठीक है. So essentially, so it's so unique. Okay. I mean, we thought we were doing evolutionary studies. से ये gene आया कहाँ से? Okay, so we were trying to see what is central to it, what is common to all the serotypes, and target those enzymes which are common to all of them. आप उसको chemically inhibit कर दीजिए. So the overall strategy was that you get rid of the synthesize होने मत दो.
Yes, sir.
ठीक है, synthesize होने मत दो. That could be a viable possibility.
Yes, sir.
Okay, that is kind of what you are kind of in a state line. Okay, now in our experience, now this is very interesting. There are 100 serotypes. Let's, for the sake of simplicity, take that each of them is 10. 10 genes are coding for one serotype. So 100 के लिए कितने होंगे? 1000. The total number of genes that Streptococcus has is 2000. The amount of, for the survival of the bacteria, the amount of genetic material that as a species, they are devoted to just the capsule thing is 50%. 1,000 genes are being devoted to making the capsule as a species. Okay, इतना investment तो किसी ने भी नहीं किया है.
Yes, sir.
Unbelievable. So all of this data is telling you that आप capsule हटा दीजिए तो गया.
Yes, sir.
ठीक है. And if you have to, and because it is so central, it has 100 genes, 1,000 genes as a species. Remember, one state has 10, the other one has 10, so into 100, which are invested at the species level to just make that one product, because it is so central to the survival of the species, and it has integrated the metabolism of the synthesis in a manner that every single pathway within the organism से कुछ ना कुछ contribution रहता है.
I'm sorry, sir. I can't take this, but I have to ask, sir. So, sir, how these D amino acids are get polymerized inside this enzyme? So, there must be an enzyme that is polymerizing these individual amino acids. So, this enzyme can be our targeted drug, sir.
See, some of these are actually.
Capsules can't be degraded.
I would tend to agree. I don't have very specific names of enzymes that we do, but some of these things for strange versions that are there are actually chemical modifications, not enzyme related modifications. So you might have to be wary of that.
Yes, sir. Yes, sir.
Okay. For example, you could have circular cyclic peptides.
Yeah, yes, sir.
Okay, those are not the way they are synthesized. Okay, so some of these are that kind of issues. On
But sir,
this.
polymerizing is entirely different. So, polymerizing a normal L is.
I understand. I'm not ruling out. I'm sure. I don't remember the name of the enzyme or which is specifically doing this. That's what I was saying. Existence of such enzymes, I will not be surprised. I just wanted to alert you to the fact that some of these things are actually also plain chemical modifications which are not enzyme driven. Okay, so that element would have to be there. Also demonstrated for many other things. जिसमें इसकी वजह से भी है. Okay, and the closest example that comes to my mind is the cyclic peptides. So similar, analogous things may be happening there, which is doing this, but that some enzyme is doing this, contributing to this, certainly possible. Given the diversity that way, but I don't. At this point in time, I remember any mistake. Any other questions people might want to have?
Excuse me, sir.
Yeah.
When the neutrophils are making net out of their chromatin, so is there a way to select that this region of chromatin they will specifically draw?
See, what we were saying is there were two kinds of this thing. One was that it is undergoing mitosis. So the cells are slowly lysing and the entire content is thrown out. Okay. The other business was a more rapid kind of so it is not necessarily selecting out. Okay, once it is induced to it's like saying that a balloon है तो आपने यहाँ से दबा दिया तो जो कुछ हवा निकल आए उसमें जो निकल गया तो निकल गया. So it is not selected portion or let's say chromosome 3, 4 and 5 are thrown out. ऐसा कुछ भी नहीं है. If you go back and take out the nets and sequence the genome, you will realize कि वो पूरा genome फिर से बन जाएगा. किसी में से कुछ था, किसी में से कुछ था. Finally, everything is there. Okay, so जो portion निकल आया, वो निकल आया. It is not within that. It is not. There is no distinction being made between this is the part or this is the one which has to be sent versus something else. So nothing like that.
So do these nets have a specific structure or just a mess?
See, now actually, nets have been shown to be involved in a variety of very diseases, and it's a whole field now. Okay, EVs is you have a journal by the name Journal of extracellular vesicle. एक पूरा journal निकला है. These nets have been shown to be important in a whole variety of situations. Autoimmune diseases, आपके पैर में arthritis होता है, वहाँ nets are been shown to be ones which are messing it up. Autoimmune diseases, a variety of other kinds. Kidney disease. If you go back and look at nets and diseases, कम से कम 5,000 पेपर आ जाएगा. And all of this work is actually moved to patients now because it's turning out that it's a, I mean, neutrophils are the first ones to show up there. People have been focusing in terms of saying, okay, उठा लिया, phagocytose कर लिया, कुछ भी, factors release कर दिया, chemotaxis में help करता है, this, that, and the other. But the fact that some of them died and released all of these is now gaining huge amount of traction and importance. So it's a huge field now. If you go back and do Google it or PubMed or something like that, it's huge. I mean whole journals are being devoted to this kind of work. There's a lot of stuff. In fact, the extracellular vesicles that were there, people are using it to cargo material. You say उसके अंदर आपने ये वाला ड्रग डाल के उसको ट्रांसफर करने में इस्तेमाल कर लिया, उसको आपने you have engineered a cell which has a certain ligand which will target a cancer cell, for example, and now make vesicles out of it. So the vesicle has these molecules, proteins on the surface which are going to, for which the receptors are there on cancer cells or any XYZ cells, and they go there, fuse, and the contents are released inside, and you can have whole variety of cargo. In it, you can have mRNA, sparkling drugs, variety of other proteins, toxins, this, that, and the other. So this is huge. I mean, it's being used as a vehicle. I mean, there is a COVID vaccine based on this now. Not in the market yet, but जिसमें उन्होंने extracellular vesicles में salmonella में COVID proteins बना के उससे extracellular vesicles बनाया. That vesicles are displaying these COVID vaccine proteins on it. So when you immunize, you get, it behaves kind of a virus minus all the pathogen components.
And maybe the proteins which are bound to the chromatin,
No,
there is some structure. It's not complex now. It is more of.
Also, to express it on the strep pneumonitis, this thing, make vesicles out of it. So vesicles are going to be decorated with this, let's say S1 protein. So you have, instead of in the virus, you have S1 with all the other khatarnak things with it. So this is salmonella. It's not capable of replication. It is mimicking the virus with all the S1 all around it. So now when you give it to the person, salmonella we anyways carry. So अब उसके साथ आपके पास ये हो गया, you are immunized with for COVID. So I mean, if you think about it as a platform rather than any particular district. So that's the big thing about it. Okay. Any other question? I mean, my email ID is here. So in case you have any questions, please feel free. And if you have more than 2 questions, come to NII.
It's not that.
Okay? Yeah.
Necrosis or neck formation is a basic process or first line we can say.
This is a basic process.
I mean, no, specifically we can say it's a first line of defense of immune system or maybe a specific. I mean, first
Specificity.
So he is asking whether it is the first line of defense or is it specific response?
Yeah. So I mean, I was.
when necrosis formation happens, self-produced stress-like condition, and stress is against.
So people are actually, see, the current state of story is that you have a pathogen, you have neutrophil. Now neutrophil has to make up its mind, phagocytose करें या नट फेंके. ठीक है, there is a whole bunch of questions around this. How does the neutrophil decide which pathway to take? And within them there are other pathways. Lyse करें, suicide type of logic, और भी से थोड़ा सा बॉम्ब फेंक देते हैं, 3 बॉम्ब में हो जाएगा. ठीक है, थोड़ा सा काम चल जाएगा तो क्यों पूरा घर अपना जला लें?
It could be the size of the foreign particle.
Whole bunch of, you go back and look at it, is incredibly interesting. ठीक है, वो आपने किया कैसे हैं? And people are doing, वो बैक्टीरिया भी नहीं डाल रहे, वो बोलते हैं यार कार्बन पार्टिकल दे देते हैं बैक्टीरिया के साइज, कोई बोलता है ग्लास देख के देखते हैं. ठीक है, कुछ बैक्टीरिया से कुछ और उसके सरफेस पे कोई और लिगेंड मॉलिक्यूल तो नहीं है जिससे ये ट्रिगर हो रहा है. नाउ PMA is the example that we had used. You बैक्टीरिया का एग्जांपल हमारे पास आ गया. अभी थिंग्स दे आर सेइंग दैट देयर आर कंपोनेंट्स फ्रॉम द होस्ट, मतलब बैक्टीरिया डैम. ओके, बैक्टीरिया व्हिच आर, जिससे कुछ मॉलिक्यूल्स रिलीज हो रहे हैं. द होस्ट, दोस आर डैम्स का फुल फॉर्म क्या है? डेंजर एसोसिएटेड मॉलिक्यूलर पैटर्न्स. ठीक है. So वो जब पोस्ट सेल्स करने लगता है, उससे उसको लगता है कि यार ये उसके बेसिस पे डिसाइड कर रहे हैं कि भैया ये इधर जाए या उधर जाए. Okay, so people are creating those kind of situations in the lab which will force it to go only phagocytosis or only this. Okay, there's a whole bunch of interesting aspects of it. Again, I mean, I think I already overshot. This is Staph aureus में वो बोला TLR2 के through कुछ होता है, it hits the same central hub molecule and which triggers this entire process. Okay, so there is people are trying to figure out the question that you just said. Here is an enzyme which may be floating around. यहाँ से signal गया, reactive oxygen species बन गया. This reactive oxygen species can also be triggered by LPS from the back virus. So the whole bunch of stuff. These are other components that are being investigated. All of them seem to get here or trigger this process. अब ये पूरा एक फंडा बन गया. I mean, one could spend a lifetime chasing what is happening. The original question is यहाँ पे at some point in time मुझे सामने वायरस दिख रहा है कि वो बंदूक निकालो या टैंक निकालो. ठीक है, so that kind of a thing. So how that fate decisions or pathway decisions are made is being looked at, and people are what we were doing is Staph aureus, but now new Piladi, somebody is looking at it this way. So it's fun.
So can we artificially use the information, like sir, after stimulating TLR?
No, that is what we did. We used PM, which is basically a chemical, but you can also bacteria से induce हो जाता है. But I mean, as I said, when we were doing it, we didn't know कि ये सब होने वाला है. This is all story 2021. Okay.
So like after giving LPS and Sapam, CSK4, like stimulators.
I think we should go ahead with NII.
For sure, he's going to do research.
Sir, it's already 1 hour 45 minutes.
One or 41.
Championship. You can take him along.
No. दिमाग कहाँ जाएगा?
Sir, I have to ask the Rajan sir that how he's doing that.
Rajan doesn't want to show up.
He wanted to join the Rajan sir lab. It was like first day he was sitting, I will not go in any other lab. I want to join that.
That's very good because science is all about curiosity. Unless you have questions, what will happen?
So the point, the idea was exactly this thing. हमने भी बतेरे सारे पापड़ बेले to figure out, and I was purposefully giving you the students' perspective कि हाँ भाई ये हुआ, वो ऐसा नहीं कि हीरा के दिमाग में आता नहीं था, वो भी आ गया, फिर वो it's like heartbreak versus sir मजा आ गया, I mean not छोटा मोटा, extreme ends. ये बोलते आज देर से ऐसे पता नहीं मेरे को रात को नींद नहीं है. And what begin the NII is because of we are worrying about कि क्या हो रहा है क्या नहीं हो रहा है. अच्छा you have to pull कैसे निकलेगा.
So, as I said, sir, as a ritual, the student has to introduce you, but then she is sick. So, I will thank you.
Okay.
So, I need to thank you. This is really an amazing lecture, and I'm sure the students enjoyed it a lot because you made it so simpler to understand, and the Hindi terminologies that you use were really, fascinating. And in fact, for me also, it was very because I'm not a microbiologist, but I really enjoyed.
I am not either.
But I really enjoyed the lecture, and hope to see you in future also for many lectures. And all of you, whosoever want to ask more questions or be more interactive, you can email sir. And I don't know many students, but I know few of the students who are doing very well. One of the student is a faculty in IIT Roorkee. So that's a asset to a PI if a student is doing well. So many of the students are doing very well.
Please visit NII. हमारे भी अच्छी कैंटीन में आता है.
NII location is a heaven in Delhi.
Please
So-
just for getting a sense of